So now we'll hear from the 2024 Edith and Peter O'Donnell Award recipient in medicine. It's my great pleasure to introduce Dr. Benjamin Deneen. He's a professor and the Dr. Russell J. and Marian Blattner Chair in Neurosurgery at the Baylor College of Medicine. After he would finish his presentation, hopefully we'll have time for one question if you stay on time. Thank you.
All right, thank you very much. This is a real treat to come and sort of close out the meeting. Thank you everybody for sticking around and bearing with me here, so I can figure out how this thing works. That's the laser. Okay, great. So today I'm going to talk about the dark matter of the brain, right? So these are astrocytes. We know that they're there. We know that they're important, but we don't really know what they do, right? This has really been a major question in neuroscience over the last probably 20 years, and we're starting to peel back some of these layers, and I'll share with you some of these layers. So here we have an image. This is a serial EM image of a neuron. You guys all recognize the neuron here. And this pink and this green cell, these are the astrocytes. And you can see a couple things stand out. Number one, they are in very close proximity to the neuron, and they're in close proximity to one another. And what I'm going to talk about today is, number one, how to make these cells. What are these lines of communication between neurons and astrocytes, and how are these lines of communication disrupted and reutilized to promote brain tumor formation? So to begin, I'll give you guys a brief overview of my lab. Everything in my lab begins with and starts with developmental biology. It's like the one thing that I truly believe in, right? And we study developmental gliogenesis, which is the generation of glial cells, of which there's oligodendrocytes and astrocytes. Astrocytes are the most prominent glial cell in the brain. And then what we've done is we've taken some of those developmental mechanisms and we've applied them to circuit function. A lot of the genes that we study in development are also expressed in mature astrocytes and actually contribute to brain circuit function and astrocyte function in the adult brain. And then finally what we do is we take these concepts of development and normal function and we apply them to malignancy, specifically brain cancer. So today there'll be two parts to my talk. The first part will be about astrocyte neuron communication. And the second part will be about this burgeoning new field of cancer neuroscience, where it's not only brain tumors, where it's also peripheral tumors get innervated, and the nerves and the neurons are essential for tumor growth. And I'll talk a little bit about those lines of communication that occur between cancer cells and nerve cells. All right, so what is an astrocyte and why do we care, right? So what's interesting is that in the central nervous system, and it depends who you ask on what continent and what mood they're in, there's roughly a 1-to-1 ratio in mammalian systems of neurons to glia. What's interesting though is that the frequency of glia increases across evolution. In worms, there's 6 neurons for every glia. In fly, there's 4 neurons for every glia. In humans and in mice, it's roughly a 1-to-1 ratio. So as you go through evolution and you acquire higher cognitive function, there's increasing numbers of glia. And there's this really great experiment that was done about 10 years ago where they transplanted human astrocytes into the mouse brain, and the mouse got smarter. I'm just saying, there could be something there, right? Okay. And so because these cells are so prominent, they execute a wide range of physiological roles. The astrocyte is a lot like the Swiss Army knife of the brain. It plays an essential role in making synapses, plays a key role in neurotransmission, it actually releases neuroactive compounds that called gliotransmission, that aids neurotransmission. They play an essential role in blood-brain barrier formation, they play a key role in metabolic support, and the list goes on, right? They do a whole lot of things. But what's important though is that they subserve neuronal function. Without the glial cell, you don't really have neuronal function, the whole circuit will fall apart, and I'll talk about that later. And you can imagine, because they play such an important role in normal physiology, that when things go wrong with an astrocyte, things go wrong with the brain. In fact, every single neurological disease either has a reactive astrocyte component to it, which I like to refer to as a distracted astrocyte, or a defective astrocyte component, where they're not functioning properly, and this causes circuit breakdown, culminating in neurological disorders and behavioral deficits. So they're really important for function, and they play key roles in disease. So against this backdrop, I'll start with developmental biology, right? So the first question I'm going to address is, how do you make an astrocyte, and why is that important? So I've spent a lot of my early career studying this process right here. And what happens early, and this is all in the mouse, right? Late gestationally, these stem cells, they're making neurons at first, they switch and they start making glial cells. This is called the gliogenic switch. And these stem cells begin to produce glia. These cells will proliferate, they'll migrate as they move away from germinal centers, and they will begin to mature. As they mature, they acquire these really complex morphology that I showed in that first picture. And what's really fascinating about this process is that it occurs in the context of neurons, right? Because neurons are produced first. So the astrocytes are being produced, right? They're migrating, they're differentiating, they're getting really bushy, and neurons are firing. So this raises really an old question in the field, and that is there a link between astrocyte development and neuronal activity? Does neuronal activity, or do neurons themselves, somehow shape astrocytes and promote their differentiation? So this is a question that one of my students asked a few years ago, and what we specifically wanted to understand is how do inhibitory neurons drive astrocyte morphogenesis? Because you can see, this is an undifferentiated astrocyte, and it acquires this very complex morphology over the course of 1 month in the mouse. So her specific question was, if we can manipulate inhibitory neurons in some way, does this influence the acquisition of these complex morphologies, right? So do inhibitory neurons regulate astrocyte morphogenesis? And to do this, she did something really cool, was she used these things called dreads, which are chemogenetic actuators. And what you do is you can inject these dreads into neurons, you can give the mice a drug, and it will artificially stimulate neurons. So what she did was she artificially stimulated neurons during this postnatal period, right? So she injected the viruses, she waited a couple days, she gave them the drug, and stimulated the neurons. And what she found is that when she activated inhibitory neurons, the astrocytes grew a lot bigger, a lot faster. They became much more complex. When she inhibited inhibitory neurons, what she found was the opposite. They didn't grow as bushy, and they slowed their development down. So what this result tells us is that the activity of inhibitory neurons is fundamentally important for the acquisition of these complex astrocyte morphologies. So she then asked another question, and that is, how are inhibitory neurons promoting this morphogenesis? What are the mechanisms that are at play? And the predominant neurotransmitter from inhibitory neurons is GABA. So she looked at we talk about data, we have a ton of data on developing astrocytes and RNA sequencing and chip sequencing and so forth. So she mined all of our data. She found that during this developmental window, astrocytes express the GABA receptor. So this sort of triggered a thought, right? So maybe the inhibitory neurons are releasing GABA, which is acting on the GABA receptor, which is doing this. So what we did was we knocked out the GABA receptor. So we got these knockout mice. We're able to specifically knock out the GABA receptor only in developing astrocytes. And what we found is that when we knocked out this receptor, the astrocytes did not become morphologically complex, right? They were stunted, they looked sick. So clearly, the GABA receptor and communication with inhibitory neurons is really important for this acquisition of this complex bushy morphology that astrocytes exhibit. And I won't go through all the gory details of this paper, but what we basically found is, again, there's these lines of communication during development where the astrocyte and the neuron are communicating. It's the activity of the neuron that is driving the acquisition of this complex morphology. And we go on to show that there's some important transcription factors that regulates expression and so forth. So we know that in development, these relationships are really important. So how about in the adult? So in the adult, I've already sort of alluded to this, where there's this line, the very close proximity. If you take a cross-section of this EM and you look closely at this astrocyte, there are hundreds of thousands of leaflets at the very periphery. In fact, it's estimated that a single astrocyte can contact up to 100,000 synapses. You try computing that, right? That's very complex. And if you go at the ultrastructural level, you can see that these leaflets in yellow are in very close proximity to a synapse, right? So they're actively listening and participating in circuit activity. So what we next did was we wanted to understand these relationships in the adult, moving from development into the adult. And so this is these are all the astrocytes in the mouse brain. You can see they are everywhere. And what we do is we knocked out these transcription factors that I've studied since I was probably a kid, like literally for 20 years since I was a postdoc, right? So we found that they're expressed in the adult astrocyte. So there's one called NF1A, which I studied forever. We knocked it out in the adult astrocyte, and we found that astrocytes in the hippocampus actually lose their morphological complexity. Here you have a control. Here is the NF1A knockout. You can see there's a clear loss of this morphological complexity selectively in the hippocampus. And when we look at the neurons, this is an electrophysiological recording of long-term potentiation, which is a physiological correlate of learning and memory. And in the endpoint knockout, you have a complete loss of LTP. This is followed up with a complete loss in learning and memory behavior. So clearly, if you disrupt the astrocyte, you disrupt the circuit, you impact behavior. So this is sort of evidence of that, and that transcription factors in astrocytes play a key role in that process. We've done the same thing with SOX9, which I won't talk about. We found the exact, very similar phenomena on the olfactory bulb. But what was interesting is that the fact that transcription factors in astrocytes are really important for neuronal function, it raised a really important question in my lab, and that is, how does neuronal activity shape the transcriptional responses in astrocytes? Because we know that transcription factors bind DNA, they move around, and they respond. But does neuronal activity influence these transcriptional responses in astrocytes? And we use the olfactory bulb and SOX9 as a model for this. So in this experiment, which I thought was really brilliant, my postdoc, Debo, what she did was she took a mouse and she just, no odor, and did a SOX9 chip sequencing. So she could see where SOX9 binds in the genome, all of its DNA binding sites. She then took a different mouse, exposed it to odor, and then did a SOX9 chip seq, and she found that the DNA binding of SOX9 completely shifts. It's night and day. In response to in response to odor and presumably neuronal activity, the astrocyte is responding. It's exhibiting transcriptional plasticity in response to the odor. So this is a really striking result. The next thing we wanted to know is, what are some of these genes, right? How is the astrocyte adapting to this odor, to this sensory perception? So she found this gene called SLC22A3, which, as you can see here in a control situation, is kind of expressed in astrocytes. When you give the mouse odor, it's really expressed in astrocytes. So clearly, this is an activity-induced gene in astrocytes. We then knocked this gene out, and make a long story, I won't go through all the gory details of the behavior, but basically we use a three-chamber preference assay. Mice prefer novel odors. When they detect an odor, they will go towards the chamber that has the odor. We knock this gene out. The mice don't really smell. They cannot detect odor. So clearly, this gene is fundamentally important for odor detection. So what is this gene and why do we care? So SLC22A3 is actually a serotonin transporter. So what is a serotonin transporter doing on an astrocyte? It has no business there, right? And moreover, why is serotonin going into an astrocyte? So this was the question that Debo then asked, is what the heck is serotonin doing inside? It's weird, right? It makes no sense until you dig deeper. So there's really great work from Ian Mays' lab who showed that serotonin actually gets added to histones. And serotonin acts as an epigenetic modifier. Serotonin regulates gene expression in the nucleus, in neurons. So we wonder, well, if that's true for neurons, maybe it's true for astrocytes. So Ian was great, gave us the antibody, and we stained, and you can see here, there is the histone serotonylated mark right there in astrocytes. So serotonin is being added to histones in astrocytes. So that raised the question, well, we have this serotonin transporter knockout. What happens to the patterns of histone serotonylation when we knock out the transporter? Is it really important or not? In fact, it is. So here we have the control. You can see here is, again, chip sequencing with antibodies against the histone serotonylated mark. In the control, you can see there it is. We knock out SLC22A3. You have a complete shift in the patterns of histone serotonylination and a massive reduction in the overall levels of it. So clearly, astrocytes, or this gene in astrocytes, is playing a key role in histone serotonylation in astrocytes in the olfactory bulb. But that raises one more question, is this at all important, right? Just because you found a phenomenon doesn't mean it's actually doing something. So we created a dominant negative form of histone that cannot be serotonylated. So this dominant negative, we can overexpress it in astrocytes. We can basically we can have it integrate into the epigenome, and it will not be serotonylated and basically block the whole process. So we did this, and when we overexpress this dominant negative histone, the mice do not, they have a deficit in odor detection. So clearly, this is really important. So histone serotonylation in astrocytes is important for their function and ultimately for circuit function in the olfactory bulb. So to summarize this work, we basically found that astrocytes are able to gate serotonin through an epigenetic intermediary during olfactory sensory processing. So giving you two examples of astrocyte roles. One is in development and one is in circuit function. Now I'm going to jump right into glioma. There's no good segue for that. So the glial nature of glioma. So I don't need to I will briefly. Glioblastoma is a universal lethal disease. It has a median survival of 15 months, 5-year progression-free survival of around 5%. It's a really, really horrible disease. It's comprised of glial-like cells, right? That's why it's called glioma. A lot of the early work in my lab looked at how developmental programs drive gliomagenesis. But what's also interesting is that glial cell function is adapted and adopted by glioma cells. They have very similar physiologies. So drawing on some of our developmental experiences, the early work in my lab focused on the embryonic chick spinal cord. And we used an electroporation model to study chick spinal cord development, where we were able to inject the central canal of the embryonic chick with DNA. We could electroporate it, and on one side, you could overexpress or knock out a gene. On the contralateral side would be a great internal control. This was the model I used probably for 4 or 5 years in my lab. So drawing on this inspiration, we developed an in utero electroporation model of glioma, where we're able to take a pregnant mouse, we can perform a surgery, the embryos will come out, we can inject the embryonic cortical ventricle with a DNA cocktail containing oncogenes or guide RNAs and knock stuff out. We can then electroporate the embryonic cortex, stuff the pups back into the mom, sew her up. The pups are born roughly 3 or 4 days later. And this is what happens at about 2 months. How do you go back? Okay. Roughly 2 months later, you get these really, really nice tumors, right? So this is a really great model, and that's immunocompetent, it's relatively cheap, it's high throughput, and it's modular. You can do whatever you want with it. So this is a great model. And oftentimes, when you create new models, it creates new opportunities. And on that note, this is where we sort of entered functional genomics, which will lead to cancer neuroscience, I promise. So this is in collaboration, as Nidhi alluded to yesterday, this is with our good friend Ken Scott, who's passed away. And really, it sort of gets to this idea of saturation mutagenesis in cancer research, where we now know roughly every single cancer mutation. There's like 3 million cancer mutations in the COSMOS database, or probably more. And we only know what like 5,000 of them do. So that's a real problem, right? So among all these mutations, which ones are drivers, which ones are passengers, decoding all of that is really important. And I'm sorry, but you can't just rely on AI to do that, right? You have to do the hard work, because that's where the data comes from, right? It's the hard work. And so 3 examples of this are NF1, P10, P53, commonly mutated tumor suppressors in all cancers. These lollipop plots show different mutations, different variants, right? There's probably 60 or 70 different variants of each of these genes that exist. Which variants are important, which variants are not important? It's a fundamental question. So we developed this barcoding system that we have combined with our electroporation model, where we can basically pair a variant with a barcode. We can do a complete allelic series of all these variants. We can overexpress them in our IEB model, and we can rip out the tumors. We can sequence for barcodes, and we can use the abundance of the barcodes as a proxy for whether a variant is a driver or whether a variant is a passenger, right? The abundance of the barcode is reflective of the constituency, and it sort of outcompetes. The other one gives you a sense of which one is a driver, which one's a passenger. So this is the system that we built, and we initiated this system on 2 dominant oncogenes, PIK3CA and EGFR. These are 2 dominant oncoproteins that, when mutated, drive a host of cancers, including GBM. And I think in GBM, there's roughly, 30 different variants of PIK3CA. Of EGFR, there's probably on the order of 50 different variants. So the idea would be to do an allelic series of each of these with a barcode and ask, amongst these allelic series of variants, which ones drive and which ones don't drive. So I'll take you through the PIK3CA example. So here's the experiment. We put in these 27 or 30 variants, each had a barcode. We electroporated them into the embryonic mouse embryonic mouse brain. When the tumor formed, we ripped out the tumor, we sequenced for barcodes, and this is reflective of the barcode enrichment. So you can see there's these 4 different variants here: C420R, H1047R, R88Q, and E545K. These were the top hits. We then went and we individually validated these in our model system. I'll have you know that some of these have up to 40 mice. That's how robust our model system is. And so you can see, basically, long story short, those that hit basically validated. It's a really, really nice system, and proof of concept that this system can screen variants. Well, that's great, but what does it mean, right? Who really cares? You found a new variant, right? Is there something interesting here? And of course there is. And like a good biologist, we sequenced the tumors, right? Because we didn't know what else to do with them. So we sequenced the tumors, and I expected them to all be the same, right? Because it's PIK3CA, they differ by a single amino acid, they should all reflect a similar biology, but they don't though. So here we have a PCA plot, basically plotting the transcriptomic data from each of these variant-driven tumors. And you can see C420R and H1047R are different from each other, and they're different from the core set. That was very, very striking. This is not something you could anticipate. You have, again, tumors driven by variants that differ by a single amino acid, generating, and they die at about the same time, at the same pace, but their transcriptomes are fundamentally different. So what differentiates them? Well, it turns out what differentiates these two are a set of synaptic genes. In fact, one of them has a downregulation of synaptic genes, the other one has an upregulation of them, right? So this was odd to us. So what do synaptic genes have to do with tumors? Like this makes no sense, right? This is like 5 years ago. And so we started thinking about, well, synaptic imbalance, right? Synaptic imbalance is often thought correlated with epilepsy and seizures, and about 40% of brain tumor patients with GBMs get seizures. So we wondered, do these different variants have different susceptibilities to glioma-related epilepsy and seizures? So to do this, we collaborated with Jeff Nobles, and we did a bunch of EEGs. So what we did was we generated basically these mice bearing tumors driven by the different variants, and we did EEGs with them. You use R88Q as sort of our base control, and we looked at C420R, H1047R, and you can see quite robustly, very early on, I think that's C420R, my eyes are really bad, or H1047R, you can see they begin to have seizures as soon as you put the EEGs on, like by P30. C420R, it seizes about 10 days later, and that is correlated with this massive increase in spikes. So you can see these brains are very hyperactive, very hyperactive. And I'll add, R88Q will eventually seize, and they will eventually have seizures just much later. C420R and H1047R had seizures very, very early on. So for some reason, these tumors are driving hyperactivity at a very, very early and a very massive, massive amount of it. It's pretty wild. So then mechanistically, what's happening? So what we found was both C420R and H1047R release something called glypican-3. It's been shown in astrocytes that the glypican family is released by astrocytes and remodels and actually forms synapses, makes synapses onto neurons. So we found glypican-3 is basically, the idea is that it's released by these tumors, travels to the synapse, rewires synapses towards hyperactivity. And in fact, that's what we found. So when we overexpress glypican-3 in our model system, you get this sort of decrease in overall survival. And tumors that overexpress glypican-3 have this really, really early hyperactivity and increase in seizure incidence. So to sort of summarize this, we use functional genomics to understand the neuroscience of brain tumors, right? And this is not something anyone could have really anticipated when we started this project. I was quite frankly bored with this project until we got to the cancer neuroscience part of it. And so we used this screening platform to find new drivers, and it led us serendipitously into this new area of sort of like cancer neuroscience and tumor and neuron interactions. So what is cancer neuroscience, and why do we care about it, right? I think it's actually a fascinating field. And really, I think what it speaks to is how tumor cells communicate with neurons. And it's not just restricted to brain tumors, but it's also relevant for breast, prostate, pancreatic, and so forth. And really, in the brain, there's 3 levels of interaction that are kind of interesting. Number 1 is direct electrochemical synaptic connections. Brain tumors make direct synaptic connections with neurons. Just wrap your mind around that. It's pretty wild. The other one is this paracrine signaling, where, as I just sort of showed, where tumors release factors that rewire neurons towards hyperactivity. Neurons also release growth factors that promote tumor growth. So it's like very Machiavellian if you think about it. The tumor is releasing factors that make the neurons more active. When they're more active, they release factors that cause the tumor to grow. It's sort of getting whatever sources they need. And then the third one is this really interesting systemic interactions where a lot of breast cancer patients have trouble sleeping. There's evidence that colon cancer can influence the reward center in the brain. So there are systemic effects, right, of cancer on this. And so, and I'll be done in 2 minutes. And so what we're really interested in here is understand these paracrine signaling. I'll be really brief here because this is really cool. So a lot of the studies in this area have really examined local interactions, how neurons adjacent to the tumor drive tumor growth. But my student was interested sort of in these remote interactions. And what he did, this is an amazing experiment. He basically put a tumor in one hemisphere with our IUE stuff, and he activated neurons in the contralateral hemisphere. And what he found is that like a moth to the flame. When we activated the neurons, the tumors moved towards the neuronal activity. So there was something about the remote neuronal activity that's driving this growth. And we did a bunch of single-cell sequencing and so forth, and basically found that there's this activity-dependent population that is found at the leading edge. That this population at the edge is specifically found when we have heightened neuronal activity. We screened a bunch of the genes from this activity-dependent population using our bar-coded system. Turns out a lot of those genes were axon guidance genes, which makes a lot of sense, right? Because chemotaxis cell movement and development appears to be utilized here. And what we found is this gene called CEMA4F, which when we overexpress it, you have decreased survival, it enhances infiltration, and actually remodels the neuron's towards hyperactivity as well. And so this sort of study sort of identifies sort of remote neuronal activity as a key driver of glioma infiltration during tumor genesis. And to summarize sort of my talk, so we talked about astrocyte development, we talked about sort of inhibitory neurons and astrocytes, sort of important for morphogenesis. You talked about brain circuits and sort of adaptive transcriptional responses of astrocytes. And finally, we talked about sort of a functional genomes of glioma and how we serendipitously moved into cancer neuroscience to study infiltration and things of that nature. And that's my lab, and I'm done. Sorry for going over.